Okay, moving on to section 5, we're going to talk a little bit about target cell specificity. We already talked about cells only respond to a hormone if they have that specific receptor for a hormone. So that is what's called target cell specificity. So one example is ACTH. This is adrenocorticotropic receptors that are found only on certain cells of the adrenal cortex. But if we're talking about our thyroxine, our thyroid hormone receptors, these are found virtually anywhere in the body, on most cells of the body. Target cell activation is going to be dependent on 3 different factors. We already talked about one, the blood levels of the hormone itself. Also, the relative number of receptors that are on or inside the target cell. The higher number of receptors that can be found either on the target cell or in the target cell means the more pronounced the actual effects of the hormone. Affinity, or the strength of the binding between the hormone and its receptor, can also affect target cell activation. So, amount of hormone can influence the number of receptors for that hormone. We have something called upregulation and downregulation. So, upregulation, the target cell form more receptors in response to persistently low levels of that hormone. And then we have downregulation, where the target cells will actually lose or shed its receptors in response to high levels of that hormone. And the purpose of shedding these excess receptors, or a lot of these receptors, is to basically desensitize the target cells so that they don't respond as vigorously to the hormonal stimulation. So basically help preventing them from overreacting because there's persistently high hormone levels. Just quickly, just mentioning the half-life, onset, and duration. So hormones, when they're in our circulation in our blood, they're either circulating freely by themselves or they have to be bound to something. So if we're talking about our lipid-soluble hormones, so steroids and our thyroid hormone, those are going to have to be attached to some sort of protein carrier. Because they are lipid-soluble, they need to have something else. All others, so our water-soluble ones, our amino acid-based derivatives, those can all circulate in our blood without any sort of carrier because they are water-soluble. The concentration of circulating hormone is going to reflect the rate at which the hormone is being released and the speed at which it is inactivated and removed from the body. So that's kind of where half-life comes in, just a general understanding of what the half-life is. So hormones can be removed from the blood by either some sort of degrading enzymes, but most of them are actually going to be degraded by our kidneys or our liver. So most hormones have to be broken down by our kidneys and liver, and their breakdown products will be released into either our urine or our feces. So if we're talking about half-life, it's basically the time required for the level of the hormone in the blood to decrease by half. And this can vary anywhere from a fraction of a minute to a week, depending on which hormone it is. But if we're talking about our 2 structural classes, the water-soluble or amino acid-based ones are going to have the shortest half-lives because our kidneys rapidly remove them from the blood. Okay, onset and duration. Hormones, again, they're very varied, so they have different response times. Some of them are immediate. Some, like most of our steroids, can take hours to days for us to have some sort of effect, and some are actually inactive until they actually get inside of the target cell itself. So they have different response times. The duration of the response is usually limited, anywhere from 10 seconds to several hours, and the effects may disappear rapidly as blood levels drop. But some of these can persist for hours if we have low concentration levels of the hormone in our blood. So all 3 of these things, the half-life, the onset, and the duration of the hormone activity are dependent on whether the hormone, again, is it lipid-soluble or is it water-soluble. So here's a good table. I recommend going in and previewing it just to kind of compare everything that we've talked about so far. and the differences between the lipid-soluble and our water-soluble hormones. Okay, the last thing I want to talk about before you actually get into hormones is a lot of times we have more than one hormone acting on the same target at the same time. So anytime we have multiple hormones acting at the same target, we have a couple of different things that can happen. One is permissiveness. That's where one hormone can actually cannot exert its full effect without another hormone being bound and being present. So a good example of this is a lot of our reproductive hormones need our thyroid hormone in order to have that permissive effect. If we lack our thyroid hormone, we end up with delays in development. Synergism, that's where more than one hormone produces the same effect on the target cell. If they're both producing the same effect, you get an amplification effect. A good example of this is glucagon and epinephrine. Both cause our liver to release glucose, and we can actually increase by 150% the glucose being released. And the last possible result, if we have more than one hormone acting at the same target, is antagonism. So one or more hormones is going to actually oppose the action of another hormone. So an example of this would be insulin, whose function is to lower blood glucose, and glucagon, whose function is to raise our blood glucose. So some of these can compete for the same exact receptor, but they act via different metabolic pathways, or even can cause downregulation of receptors, like we talked about before, some sort of shedding for the antagonistic hormone. Okay, so that's it for sections 1 through 5 for 16. I do recommend that you kind of briefly go through the slides to see if there's any pertinent information for the remainder of this chapter. But the bulk of the hormones, the hormone functions, and what happens if we have hypersecretion or hyposecretion is all going to be in that supplemental non-PowerPoint lecture that I have. So definitely make flashcards or do whatever you need to do, or use those hormone cards that I already have made for you. You just need to print them and cut them up to help you study for these hormones.